What if aging isn't destiny, but a program we can rewrite? What if the same cellular instructions that drive degeneration could be reset safely, precisely, and at clinical scale? And what if I was to tell you that the first real test of this idea in humans is happening now? Today's guest is helping lead that transition from epigenetic theory to the first ever partial reprogramming candidate poised for human trial. Welcome back to Decoding Longevity. I'm your host, Raghav Sehgal, and I'm joined with my co-host, as always, Dr. Maxwell Jacques.
Hi, everyone. In our previous episodes, we have explored everything from immune aging to functional decline. And today, we take a clinical translational turn, talking to someone at the operational helm of a company that is advancing cellular rejuvenation therapies into human testing. Our guest today is Dr. Michael Ringel, Chief Operation Officer of Life Biosciences, a biotech on a mission to reverse and prevent multiple diseases of aging by targeting the biology of aging itself. Michael, welcome to the podcast.
Thank you, Maxwell, and thank you, Raghav. What an amazing platform. I love your podcast. I love the work that both of you do. And I'm really excited to share the great news of what's happening at Life Biosciences.
Yeah. Well, Michael, we're really excited of what you're doing. But first, I want to talk about your background and your journey into sort of the space of biology of aging, which, by the way, is such a cool journey. So your background sort of spans biology, law, and decades of biotech strategy. You started at BCG, Boston Consultancy Group, before joining Life Biosciences, almost spending, I would say, 25 years. Is that correct? Right? But how did you sort of come to focus your career on aging biology and sort of translational longevity science? And why become the CEO in this rapidly evolving field?
Yeah, it's a great question. Obviously, it's a topic that's of interest to everyone because everyone is aging.
Yeah.
I remember even conversations with my father about it, when I was young. My sort of professional journey starts when I am a trained biologist, undergrad, Ph.D. Then, as you talked about, I spent 25 years at BCG, Boston Consulting Group, where my work was really heavily focused on biopharma, particularly R&D, and how we can make progress against the many diseases that affect us.
Yeah.
And one of the projects I did was for a client scanning the landscape of longevity technologies. It was a great project, led them to some excellent action in terms of building some philanthropies around this. But it also led me to, take part in that landscape assessment of where we thought the most promising technologies were. And I just completely fell in love with partial epigenetic reprogramming, which is a bit of a mouthful, but incredible science that obviously we'll get into during the conversation. And I just became convinced that it was the right move to bring this forward, and I wanted to be part of that journey. So I called up the company and asked them if I could join it, and here I am.
Yeah, no, that is incredible. It definitely is hard not to fall in love with the longevity space and the research, especially if you having a great life and you want to be here for longer, right? So it's definitely something that also drawn me to all of this area. But okay, so for our listeners that are new to Life Biosciences and to the partial epigenetic reprogramming idea, could you maybe explain a little bit what is actually that and what is the company's mission and the foundational idea behind this new approach, and how is that different from the other traditional approaches for age-related diseases?
Yeah, a perfect question, right? You're really hitting on the core of what is this. It starts with the fact that probably everyone knows babies are born young, right?
Yeah.
I haven't told you anything you don't know yet. But babies are born young, but every baby starts as a single old egg cell and an old sperm. And those cells are old. You can measure that they're old. They're old functionally, they're old transcriptionally, they're old epigenetically. And in fact, even after fertilization, the zygote stays old for a few days, which is sort of amazing to think about. At the very start, babies are actually still old, and they're not babies yet, right? It's just a fertilized embryo. But during those first few days, it's old. And then between day 7 and 9 of embryogenesis, a miracle happens. And after day 9, if you measure the age of that zygote, it will measure 0 on an epigenetic clock and close to that on a transcriptional or functional clock. And what happens between day 7 and 9, we know, thanks to some work out of the Gladyshev lab and others, what's called the epigenetic marks. These are chemical groups that are on the DNA and on the histones the DNA is wrapped around. So methyl groups, acetyl groups, and so on. And during this period, they're actually wiped back to kind of the initial slate that a baby would have. So there has to be a process in nature that completely rejuvenates a cell. Otherwise, you couldn't have babies that just come from old cells. And that's actually been tapped into artificially. When we make a clone starts as a single old cell from an old individual. You can take the skin cell from an old mouse. And Shin Yamanaka won the Nobel Prize in 2012, showing you could treat that cell with just 4 small proteins, abbreviated OSKM, and take that cell back to a state where it's essentially young again. And then when you grow the clone from it, the clone is born a baby, and then it has a normal life. It can, have normal pups of its own, etc. In the case of a mouse. So the thing that people sometimes find conceptually hard to believe, that we could completely rejuvenate, we actually already do it at the single cell level in the context of making clones. We totally rejuvenate the cell. And of course, we're just tapping into a mechanism that already exists in nature that makes babies young. Now, the issue is you can't do that in your own body because you don't want to take your cells all the way back. Like your nerve cells have to stay nerve cells. Your muscle cells have to stay muscle cells. Skin has to stay skin. But the insight that came out of the Sinclair lab at Harvard Medical School in 2020 and was on the cover of Nature magazine is that you can treat with just 3 of those 4 proteins, abbreviated OSK, without the M, and you take cells only partway back. They stay what they are. They keep their identity, so it's safe. And what they showed in that study is that you could give lifetime administration of OSK and completely safe. In fact, what they saw is it decreases the rates of cancer in that study. Other labs have treated lifetime with OSK and found, the only side effect is it makes mice live longer, right? So safe, but profoundly therapeutic, because when you can de-age cells, they can heal themselves. And in that first study, what they showed was mice that were blind from 3 different causes. One was a glaucoma model, where you insert microbeads to kind of increase the pressure. The second was just aging. Visual function decreases with age. And then the third was a physical crush model, where they crushed the optic nerve with forceps. So blind from 3 different methods, in all 3 cases, able to completely restore vision, not just to that of an old mouse, but to that of a young mouse. So profoundly valuable, obviously, in mice in that first study, but it points the way to what could be done in other species, including humans, not just in the eye, but in other organs as well.
And so that's really fascinating, Michael. You've really sort of talked about this journey of how sort of this has really evolved as a field, right? And we've gone from sort of the initial understanding of the Yamanaka factors, right? Dr. Shinya Yamanaka figured out how sort of these 4 factors could really sort of go from any sort of cell to the original sort of stem cell-like state, or the original stem cell state, and that got him the Nobel Prize. And then sort of Dr. David Sinclair and a couple of other folks figured out we could essentially do this idea of partial reprogramming, right? Which is sort of not go back to that whole stem cell state, but sort of this intermediate state, which is perfect to do this in the body. But can you sort of explain to our listeners that why is it so important that we do this partial reprogramming in humans, right? Why can't we do this full reprogramming in humans? Why is it okay why can't we do the whole OSKM cocktail, right? As we call it, as Yamanaka discovered. It's a Nobel Prize winning this thing, right? Why not do the whole mix?
Yeah. So a number of labs now have looked at OSK, which is the combination we're using. We've also looked at it in a number of preclinical models. There's never been a published study showing a safety issue from OSK. We've never seen it in any of the work we've done. Multiple labs have shown if you treat with OSK, it makes mice live longer. Multiple labs have shown if treating cancer cells with OSK causes them to apoptose, would kill themselves. So reducing rates of cancer consistent with the findings in that first study that showed, no increase in tumors and, if anything, a small decrease in tumor frequency. So there's a quite large already and growing body of data suggesting OSK is safe in these preclinical models.
Yeah.
That's been supported not just in mice studies, but now in non-human primates, monkeys, which of course are more similar to us than mice. Prior to approval to start our phase 1 trial in humans, we did studies in non-human primates with intravitreal injection in the eye, which is the first indication we're going in humans, and checked for safety issues, because obviously you want to make sure drugs are safe and effective. And half of that equation is the safety. And what we saw in the non-human primate study, which was then reviewed by the FDA, were literally zero significant adverse events from the administration of OSK. We did see some small administration from the issues from the vector, the way you deliver the OSK, some localized inflammation, which is as expected, because we'll talk about it, I'm sure, later, but AAV vector is known to cause a little bit of inflammation. It's already on the market in the eye, so it's not an unusual approach, but we know that it can cause that. So we did see that, but we saw no issues from the OSK itself. But we also saw incredible efficacy, the same as what we saw in the mice, where we saw restoration of vision, even with very small sample sizes. We had 4 and 6 monkeys per treatment group. What we saw was a statistically significant increase in visual function. And, this is in a pretty profound injury model. So again, consistent with the hypothesis that in humans we'll be able to restore visual function in a safe manner. Of course, now we have to run clinical trials to assess that. And that's a staged approach where you start with phase 1 trials, small number of patients, and then expand from there if successful. And that's the stage we're at now.
Yeah. I remember reading, David Sinclair's paper with the mice and restoring the whole vision. And I remember, oh my gosh, just blow my mind out. I was like, okay, now we can actually restore everything back. And since then, from 2025 to 2026, we have seen Life Bioscience not just generating, but gearing up to bring this partial reprogramming candidate to human trials. So this is quite incredible. So this is a program called and led ER100, so targeting optic neuropathies, and that's a huge step. But can you tell us a little bit, where does this trial stand? You just mentioned that it has been done, the safety, so I assume that's the phase 1 done and dusted, I suppose. And if you're talking about specificity and efficacy, that's phase 2, phase 3 trial. So where are we at? How long are we still going to take to have this available? And why do we have to do it targeted? Why can we not just do something that is completely systemic? Why does it have to be injected and potentially not just ingested and have a full partial reprogramming?
Yeah, great question. So I'll split that in two. First is talking about ER100 and the program in optic neuropathies. And then I'll talk about the second part of your question, which is other diseases that could be amenable to treatment with partial epigenetic reprogramming. So ER100, as you rightly mentioned, was granted in IND authorization. So the ability to test in humans in just a few weeks ago by the FDA. So we will be starting our phase 1 trial shortly. Phase 1, I think many people know, but probably not all of your listeners know, is a trial designed to assess safety and tolerability. Typically then in phase 2, you might assess the right dose level. And in phase 3, you would do a large study with many patients to get a statistically significant set of data on whether you're having an efficacy impact. So phase 1 is generally designed for safety and tolerability. We are actually dosing patients, which is unusual in phase 1. But in discussion with FDA, we were granted the ability to do that. That means we will be treating patients who have optic neuropathies. I'll explain what those are in a minute. But in addition to our safety and tolerability endpoints, we will also gather secondary endpoints on efficacy. It's a very small number. We don't expect a statistically significant signal with a very small number of patients, but we hope to get some anecdotal data. And the kinds of things we're measuring are the very standard clinical parameters that you would measure for is visual function improving or not declining as fast as it used to decline. Things like what's called best corrected visual acuity. That's basically when you take an eye chart, how far down the eye chart you can read. Things like Humphrey visual field, which is an assessment of how good your peripheral vision is. And there's some other measures we're doing as well that we think are maybe even a bit more sophisticated to really measure, is this working in addition to the safety and tolerability? But the real focus of efficacy, is it working question will be in the later phases. Now, why optic neuropathies? We're going after two related diseases. One is called open angle glaucoma. Many people have heard of glaucoma. It's the number one cause of blindness in the elderly. In many patients, it starts from increasing what's called intraocular pressure, the pressure in the eye, but not in everyone. Actually, there are some people who get glaucoma even without elevated IOP. But eventually damage happens to the nerve cells of the eye, what's called the retinal ganglion cells.
Yep.
The other disease we're addressing is called NION, non-arteritic anterior ischemic optic neuropathy. Most people have heard of that, but it is actually the most frequent acute cause of blindness in the elderly. It's similar to a stroke of the optic nerve rather than a stroke in the brain or a myocardial infarction in the heart. So it's caused by lack of blood flow, but the ultimate result is the same: damage to the nerve cells of the eye, these retinal ganglion cells. And so if you can rejuvenate retinal ganglion cells, regardless of the upstream cause of the damage, you may be able to ameliorate the disease. And of course, it's hugely valuable, obviously, for people with blindness to either halt the progression of the disease or slow the progression or potentially even reverse the disease so you have some restoration of vision, as we saw in the non-human primates and in mice. That's incredibly valuable, both for the blindness itself, but also blindness leads to a lot of other issues. It can cause falls, which are obviously very devastating in the elderly. It can lead to a lot of isolation, and that can cause mental illness, mental decline, also neurodegeneration rates. Things like Alzheimer's are elevated in people who have blindness because of this kind of isolation. And so obviously blindness itself is a large unmet need, but it also leads to a lot of other conditions that obviously are very severe. So there's a lot of value in addressing just this first set of diseases, which we're hopeful will have good data in as we go through clinical trials. Your second question, though, is, what else? And there are lots of other diseases. We think, in principle, every age-related disease, which is more than 90% of what causes mortality in the developed world, should be amenable to this partial epigenetic reprogramming approach. It's really a broad platform. And there's a lot of data now to suggest that these epigenetic changes happen in a whole host of age-related diseases across organ systems. And there are a number of publications showing reversal of age-related disease in other organ systems. One just recently came out in the journal Neuron, showing in a mouse model of Alzheimer's disease, which of course is an imperfect model for human Alzheimer's disease, but in a mouse model, treating with partial epigenetic reprogramming, OSK, led to a restoration of memory and cognitive function. We recently released data showing in the liver that we were able to restore function and ameliorate disease, again in a mouse model, in a model of a disease called MASH, which is basically fatty liver disease. It's one of the most common serious diseases in the developed world, tremendous unmet need. And what we saw is that the same approach, OSK, delivered through a different vector and a different promoter, but the same underlying mechanism, rejuvenated cells in the liver and led to reduction of many of the parameters of fatty liver disease in this mouse model. So there's a lot of evidence that this is a platform that can be used broadly across age-related diseases. But of course, you have to show the evidence. You have to run the clinical trials, test these diseases in humans, see if what we see in these animal models actually also works in people.
Let me just ask a quick question, piggybacking on the answer that you gave. So obviously there is the potential for all of those diseases, and there is a lot happening, and there is so many models that have shown success. But could you tell me, why do we need to do one at a time? And can we not do systemic partial reprogramming? And is the only way that we can reach or deliver those vectors via injections, local injections? Or is there, I hear talks of, maybe potentially just taking a pill that can induce partial reprogramming. So could you talk a little bit about that?
Sure. So in the mouse model of fatty liver disease that I described, that's a systemic administration. We administer via an injection that is a whole body injection, but it has a particularly specific vector that hones in more frequently on the liver, and a promoter that turns it on only in liver cells, ideally. Of course, we'll need to assess that with more data on exactly how specific we are, but you can do systemic delivery that then goes to specific organs. And of course, that's important because what we're aiming to do with this technology, at least in the near term, is address age-related diseases. So there are many, many age-related diseases that affect us. Obviously, they affect us more as we age, in fact, go up exponentially with age. And again, more than 90% of mortality in the developed world is some kind of age-related disease. And so if we can address some of the biggest ones first, but of course, hopefully all of them with time, that's a way to actually ameliorate a lot of human suffering and create a lot of value for patients. And so our approach is a very standard biotech model to address disease. We're not looking for regulatory change or any kind of different way of how regulators think about this. We want to show we're benefiting patients who have disease and improving their lives. We want to do so in a very rigorous clinical trial approach. What's different is the underlying mechanism is one that is rejuvenating their cells, but the endpoints, the kinds of things we measure to see are we actually making their lives better, are very standard.
Yeah, no, that's really interesting, Michael. I do have one follow-up question before I ask my final question from you, which is around sort of the choice of why Nion. Right? Was this a strategic choice, given that we're seeing potentially an increase in Nion, given the rise of sort of GLP-1 intake and Ozempic intake that has been sort of reports around anywhere between 2 to 4 times more likely in cases of Nion of people taking Ozempic, and that would only rise as the consumption of Ozempic and GLP-1s increase. So was that the strategic decision to take sort of Nion as one of the conditions versus glaucoma, which is clearly a diabetes-related condition, which might actually go down as consumption of Ozempic?
Yeah, so you're referring to, there is evidence in the literature of an association between the use of GLP-1s and an increase in risk of Nion. These medicines are still incredibly valuable, right? They drive a lot of patient value. It's outside of my scope to, sort of talk about, when anyone should take or not take those medicines. I think, consulting with a physician, there are ways to assess your risk of an event like Nion by looking at things like the cup to disc ratio in the eye, which is a kind of relatively routine eye exam to understand, could you be at potentially elevated risk? But, that's obviously a topic specifically focused in an area outside our remit. It was not an input to our decision. We decided to go after Nion before there was anything in the literature around this potential association. The underlying reason why we focus on Nion is that it's a very similar downstream problem to glaucoma, as in it's damage to the retinal ganglion cells. It has a different upstream cause, whether it's an ischemic event, a lack of blood flow, or a pressure-related event, as it could be in many glaucoma patients, or kind of a different event in patients who have damage from glaucoma without elevated pressure. Ultimately, different upstream causes all driving damage to the downstream part we care about, which is damage to the retinal ganglion cells, that we are hopeful, and of course it will be up to the clinical trial to prove it, but hopeful that we can ameliorate that damage by rejuvenating these cells.
That makes a lot of sense. And yeah, it's amazing that this was sort of the choices you made in making this decision. I guess my final two questions to you are sort of forward-looking in a way. So looking ahead 5 years, so what does a successful longevity biotech ecosystem look like for you?
Yeah.
Beyond life sciences sort of having its first clinical candidate, which, fingers crossed, will be there. What breakthroughs do you think are on the horizon? What hurdles do you think still need to be cleared? And my second broad scope question is, who do you think we should bring on this podcast to really disseminate knowledge? Actually, James suggested that we should bring you, and that was how we got connected to you. So we would love to know who should potentially to really disseminate longevity science.
Yeah, great questions. I mean, so I'm really hopeful that beyond just our own platform, the insights that we can glean from aging biology will help us address a lot of age-related diseases and ameliorate a lot of human suffering. Again, we know that most of the diseases that afflict us are age-related. The number one risk factor for them is age, how old we are. And if we can do things to manipulate aging biology, we may be able to ameliorate a lot of different diseases. There's a lot of room for multiple companies in doing that, right? This is not a competitive game. The only competition is disease, and that's who we're trying to address, right, and help people. And I'm very hopeful that a number of companies will be able to be successful in doing that. There are lots of great names in this space. I actually think, my colleague Sharon Rosenzweig-Lipson, who's our chief scientific officer, would be great. I think your audience is a relatively sophisticated scientific one, and she can go, quite deep in terms of thinking about what are the mechanisms here and what we can see in the clinical trials. So, but there are lots of other people. Certainly, I wouldn't restrict it just to her. Outside our company, within our company, that can really help proselytize the potential benefits to people by working in these areas.
Yeah, no, this has been really, really insightful. I'm so excited for everything that is happening and it is to come. And as you said, like the only adversary here is really disease or aging itself. So, when someone wins in the field, everybody wins. So I think this is great about what we do. So thank you so much for coming into the podcast and to bringing all of the insights around what Life Biosciences is doing and the new therapies and how we can actually now not only think about avoiding disease but actually reversing back some of them as well. So thank you so much for bringing light for us today.
And to our listeners, we will link Life Biosciences science, clinical pipeline information, and some of Michael's prior talks, which, by the way, you should check out some of his talks from ARDD, I think 2024. They're amazing. I personally love it, in our show notes. And if you enjoyed this episode of Decoding Longevity, don't forget to follow, share, and leave us a review. Thank you, Michael, for tuning in, and thank you for our listeners for tuning in. We'll see you next time on Decoding Longevity.